Good afternoon, and welcome to the UCSF Academic Senate's 20th Annual Faculty Research Lectureship in Clinical Science. I'm Vanessa Jacoby, professor in the Department of Obstetrics, Gynecology, and Reproductive Sciences, and vice chair of the Academic Senate Committee on Research. It is my honor to introduce the Academic Senate Faculty Research Lecture. This award recognizes the exceptional career achievement and major contributions of faculty in clinical science research. Nominations are made by UCSF faculty who consider the research career of their colleagues and submit nominations for this prestigious award to the Academic Senate Committee on Research. Every year, the committee then selects a recipient to highlight and celebrate their achievements. Today, we are very proud to honor Dr. Esteban Burchard, who will be introduced by his nominator, Professor Tejal Desai, and his longtime friend and collaborator, Dr. Dean Shepard. Please note that this event is being recorded and it will be available on the Senate website. There will be an opportunity for questions and answers at the end of the lecture. So please submit your questions and comments using the Q&A function at the bottom of the Zoom window. And now to introduce Dr. Desai, who is the Deborah Cohen Endowed Professor in the Department of Bioengineering and Therapeutic Sciences in the Schools of Pharmacy and Medicine. Professor Desai's research spans multiple disciplines, including materials engineering, cell biology, and tissue engineering. Dr. Dean Shepard is a professor of medicine, chief of the Division of Pulmonary Critical Care, Allergy, and Sleep. His research focuses on the molecular mechanisms underlying pulmonary fibrosis, asthma, and acute lung injury. Dr. Desai.
Thank you so much, Dr. Jacoby. It really is such a pleasure for me to be here today on this really special evening or afternoon, and especially gives me great pleasure to introduce Dr. Esteban Burchard as the recipient of the Academic Senate's Faculty Research Lecture in Clinical Sciences. Dr. Burchard's scientific achievements have had immeasurable impact on UCSF and the lives of so many people across the globe. I'm just going to spend a few moments talking about some of his contributions, which are really so numerous to highlight right now. But he is internationally recognized for his contributions to our understanding of the genetic basis of population differences in pulmonary diseases, in drug response, and the critical interactions between genes and the environment that really drive health disparities in this country. He's been a tireless advocate for increased research to address health disparities and is one of the leading voices nationally and internationally for the inclusion of diverse populations in human genetics. His research is highly collaborative and he is an outstanding mentor, really bringing together a diverse group of trainees who reflect his ethos of doing science with impact. And his lab really reflects the type of diversity that is the goal for UCSF, and one that can really have a broader impact in the biomedical research enterprise. A couple of examples just to give you a glimpse on his impact. Intrigued by the varying responses of Puerto Rican and Mexican asthmatics to albuterol, Dr. Burchard established tools to analyze populations to really tease out the impact of ancestry in various populations. He was the one who pinpointed, through meticulous research and a body of work encompassing over 285 peer-reviewed publications, the specific genes responsible for specific responses, and showed that these ancestries affected people's responses to current therapeutics. And most importantly, he really showed that failure to acknowledge these differences emanating from racial disparities could result in misdiagnosis and imprecise treatment. Importantly, Dr. Burchard has made it his mission to eliminate sampling bias, which has life-changing consequences. In a Nature article he co-authored in 2011, he challenged the NIH for its lack of compliance with its own 1985 mandate for the inclusion of diverse subjects in its studies. He was able to really open the eyes of NIH and many institutions around the country to show how important it was to include these diverse perspectives. And in 2021, he published a piece on race and ethnicity and its reckoning in the New England Journal of Medicine. I could go on, but one thing is clear. Dr. Burchard has fought for racial equity and precision medicine for decades, and he has shaped healthcare as we know it today. That is why I'm so pleased and thrilled that the depth of his scientific achievements and the impact on all communities is being recognized here today. And to share a little bit more about Dr. Burchard's journey and what brought him here to UCSF, I will turn it over to Dr. Dean Shepard.
Thanks so much. It's really also an enormous pleasure for me to contribute to the introduction of Dr. Burchard. So Esteban's a San Francisco native who grew up in the Mission District and was raised as one of 4 siblings by his single mother. So he's had a mixed relationship to fishing throughout his life. So this is a picture from Mission Rock in 1975 when I think Esteban was 8 years old. And his dad, who was only at home until about this time, used to go to Mission Rock Resort to drink and would leave Esteban sitting on the rocks fishing by himself. So he had a pretty mixed view of fishing early on, but he certainly embraced fishing over the subsequent course of his life and has found fishing actually to be a source of major achievement and gratification and also fun. So his mom helped with that when at the age of 12, she arranged for him to work as a junior deckhand on San Francisco fishing boats. And she really did that because she was worried about Esteban being on his own while she was working in the Mission District in the summertime. And that paid off, but he was still having a tough time of it until he had the good fortune to be expelled from St. Ignatius High School as a freshman and landed at McAteer High School in San Francisco, where there was a dynamic young wrestling coach who took Esteban under his wing. And wrestling has actually provided for Esteban, as it did for me, an object lesson in the value of hard work, and perhaps most importantly, an opportunity to build self-confidence. He subsequently parlayed wrestling and quite a bit of intellect, grit, and determination into a college degree from San Francisco State, an MD in his first scientific training at Stanford Medical School, and then a residency at Brigham and Women's Hospital in Boston. And from his first experience with research, Esteban knew that he wanted to focus whatever research he did on work that would have an impact on the dramatic health disparities that characterize US healthcare. And he found the opportunity to do that as a resident when he connected with Jeff Drazen on a study they published in the New England Journal before he came here as a fellow, showing an impactful sequence variant in the interleukin-4 receptor gene that was greatly enriched in African-American patients with asthma. But in a preview of the rest of the pattern of the rest of Esteban's extraordinary research career, he quickly changed the direction of the project and began digging into the genetic underpinnings of the dramatic differences in asthma prevalence and mortality between 2 different groups that were traditionally lumped together as Latinos, Puerto Ricans, and Mexican Americans. As somebody who really understood what that term meant, Esteban was really able to provide enormous insight that Jeff and others involved in the project really didn't have. So we were incredibly fortunate when Esteban decided to return to San Francisco for his pulmonary fellowship. And really quite amazingly, he brought with him a substantial amount of NIH funding, which I've never seen another entering clinical fellow do. And it was based on a grant he'd written together with Jeff when he was a resident to study the genetic underpinnings of the differences in asthma mortality and severity between Mexicans and Puerto Ricans. I was especially fortunate that Esteban chose me as his alleged mentor. But as many of you know, Esteban's a force of nature, and he knew exactly what he wanted to do. So my major roles really were as a cheerleader and occasional facilitator. And so Esteban knew that he needed to build a large network that nobody had built before, with sites all around the country and eventually around the world, in order to be able to enroll enough patients to really study in detail the genetic underpinnings of this major difference in asthma mortality and severity. And within a few years, in his spare time as a clinical fellow, he built the largest cohort of patients that could be used to study genetics of asthma in Latino Americans. And not being satisfied, because Esteban never is, he rapidly turned around and also built the largest cohort in the world focused on genetics of asthma in African Americans. And as Tejal's already alluded to, these cohorts and the meticulously compiled clinical databases and biosamples he oversees have facilitated a continuously growing series of remarkable biologic insights by Esteban and also by his collaborators around the world. So Esteban continues to build new collaborations, write impactful papers, and receive an extraordinary number of large NIH grants. And this has led many of us to wonder whether Esteban ever sleeps, but we finally now have definitive evidence that he does. So finally, as Tejal alluded to, perhaps Esteban's most enduring legacy will be his fierce and unwavering advocacy, calling attention to the needs of populations that have been historically ignored by the biomedical establishment. And his commitment to diversity and equity is the engine that drives his remarkable research productivity. And also, as Tejal pointed out, he's from the beginning of his academic career, he's authored a series of very high-profile papers, beginning with a 2003 paper in the New England Journal of Medicine, calling attention to inequities in medicine and important inequities in research funding. So I expect we're going to hear more in this talk today about the strong connection between Esteban's research and his advocacy, and I'm really looking forward to his talk.
Great. Thank you, Tejal, and thank you, Dean. Let me set up my slides. Okay, can you see that okay?
Yes, you're good.
Great. Well, first of all, thank you for the wonderful introduction, Dr. Desai and Dr. Shepard. And I want to thank the Academic Senate for selecting me. My name is Esteban Gonzalez Burchard, and I'm going to talk about my wonderful journey from the hood to academic medicine. Today is Dia de los Muertos. It's also called the Day of the Dead. It's a Mexican holiday in which we invite our ancestors back for a reunion. It's actually a celebration. And on the left, I have a couple that's celebrating in Mexico. And on the right, I have my personal altar at home. And what I'm going to use this for is to acknowledge some of the people who have been very influential in my life and who have subsequently passed in the last couple of years. So the big debate in medicine is whether or not we should use race and ethnicity in clinical and biomedical research. Well, the world has been divided into two camps. One believes that it's a social construct, while the other believes it's a biologic construct. But individuals like me and many others at UCSF and around the nation believe that it's a complex interaction and not this black or white dichotomy. And I was actually shocked by an email that Josh Adler, who's the clinical officer of UCSF, chief clinical officer of UCSF, he gave me permission to publicly share this email. But in this email that he published on October 14th last year, they announced that they are excluding race and ethnicity from estimates of kidney function because it could perpetuate the incorrect belief that race has a biologic basis. Well, unbeknownst to Josh, I've been thinking about this all my life. And this is a picture of my grandfather, who's second from the right, Esteban Gonzalez. We are Mexican. He was a farm worker in the Central Valley. And when I took this photo, I didn't realize that it was superimposed on the top of my current issue of the New England Journal. So I'm very proud to say that I'm descendants of farm workers. My mother was a farm worker, and now I'm a full professor at UCSF. This is my extended family. These are my great grandparents and my grandmother, and my grandfather's on the top right. But as you can see with my great grandfather, we are racially mixed. He's part African, he's part Native American, he is part European, and that's what makes up a typical Mexican. This is my mother, and you can see that she's very dark and I'm very light. And I recall the days when I used to crawl in her bed and she used to read the Dr. Seuss books to me. And one of the books that she read was, "Are You My Mother?" And that question has always challenged me throughout my childhood and my current existence. But I was very fortunate to get a 23andMe on my mother before she passed away. And these are the results. On the left are the results of my mother's ancestral composition. On the right are of my ancestral composition. And you can see that my mother is 53% Native American. She's about 39% European and about 5% African. I'm 28% Native American and 65% European, and the rest is African American ancestry. And these early discoveries or recreational sciences have really served the foundation for my future work. But before I dive into my science, I want to talk about a little bit more about my background. Growing up with a single mom, my mom had to have a distributed network of women who took care of me. And so one of the women that took care of me was Alicia Joe, shown here. And she's in my house now watching Zoom in the other room. She's Chinese and she had two young boys, Michael on the left, Marco on the right. And in my formative years, all the way up to 13, I spent probably more time in San Francisco, Chinatown than I did in the Mission District. I even went to Chinese school, Cantonese school for 2 years. Unfortunately, Marco died in 2016, 5 days before his 49th birthday of an adverse drug reaction. So as I mentioned, I was born and raised in the Mission District. And for those of you that don't know, the Mission District is a Latino barrio in San Francisco. It's well known for its great food, great weather, vibrant culture, but also violent gangs and drugs. And as a fatherless kid, I was susceptible to all those. And as Dean mentioned, I got kicked out of my first high school. I was bused from an all Latino neighborhood on the east side of San Francisco to the west side of San Francisco to an all white magnet school. And they let me know that I did not belong. I had fights every day and ultimately got kicked out. The picture that you see here is a picture of me in Balmy Alley. On the left is a mural of mothers who have all lost their children to gun violence. In high school, my best friend was shot and killed, and I knew that I was headed down a path unless I changed. I was very fortunate to get kicked out of SI because I landed in inner city high school that had a very rigorous wrestling program. The program taught me discipline. It taught a lot of young men who didn't have fathers discipline. It's like joining the military. We lost weight. We had grit. We were in great shape. We went to the California state meet. We had some state placers. But more importantly, I went on to go on to college and wrestle. And there I met some wonderful mentors. And this is my coach, Dr. Morris Johnson. You can see he's African-American. He was an NCAA champion, two-time Olympic team member in '84 and '88. And he was also pursuing a PhD. He was a father figure to me. He was a tremendous role model. But the team that I was on, we were national NCAA champions, and we attracted a lot of individuals from around California. And it's the first time in my life that I met positive role models that were national champions, that were Olympic champions, that were doing something positive. And it's quite a contrast to what I experienced growing up as a child in the Mission District. Well, my mom was a school teacher, and she was also an activist. And so growing up, these two individuals were embedded in my life, Dr. Martin Luther King and Cesar Chavez. And in fact, they served as a moral compass, if not the North Star, for everything I did. I was fortunate to go to a great college where we had very strong biology program. And I was fascinated by biology and math, but I wanted to have clinical applications. And in particular, I wanted to meet in this sweet spot of the Venn diagram. And I wanted to go to medical school to do medical research, but I knew that the odds were against me. And this is a slide that was recently published in the New England Journal this year, 2021. And it describes who gets into medical school over the last 43 years. The top graph shows whites and Asians, and women are on the left, men are on the right. You can see that women have steadily increased. White men have decreased over the last 43 years. Asians and South Asians have steadily increased. Now, if you look at the bottom two graphs, those are the graphs for minorities. But don't be deceived, because the y-axis is truncated. Instead of comparing 0 to 100, we are now comparing 0 to 4. And you can see in the lower right corner, the number of Black men in 1978 was 3.8, down 2.9%. And if you account for US-born African Americans, that number would almost be 0. Latinos are the same. And I could talk for hours on this slide, but the main point that I want to emphasize to you is that my entire academic career, from medical school, residency, fellowship, to faculty, to even now, has been a lonely existence. I'm often the only minority in the room. I'm the only one here that's doing minority research. I'm often second guessed. I'm often put down. There's macroaggressions. There are microaggressions. And I want to flip the tables. If this were the case for women, we would be up in arms. This was the case for women 50 years ago when medical school classes had 1 or 2 women. Today, medical school classes have 1 or 2 minorities, and it's a lonely existence. So I was very fortunate to get into Stanford. And in fact, I got into a medical scholars program, which is like an MD, PhD light program. But Stanford is a world away from the Mission District. And I want to show you that we had a rude awakening. On my first medical school exam, Rob Dodd, who's featured on my right, and I were pulled over by the Stanford police while driving to our first medical school exam. We were pulled out of our car, we were frisked, we were interrogated. What are you doing here? Why are you here? Where are you going? And anyone that's been to medical school can tell you that medical schools or exams are very stressful enough. And to be late was terrible. And this feeds right into the social determinants of health. And it reminds me of an event that happened 26 years earlier with the civil rights workers, James Chaney, Michael Schwerner, Andrew Goodwin, who were arrested by the police handed it over to the KKK, and killed. And although we were not killed, it severely impacted the beginning of my medical career at Stanford. Rob Dodd is now an MD, PhD, and a neurosurgeon. And if you have the misfortune of having a brain tumor and are at Stanford, Rob's going to operate on you. I was an ICU physician at San Francisco General. And when the President of the United States came to town, the FBI interviewed me preemptively to make sure that if the president came to my ICU, I would not kill him. But I'm telling you that to show you the treatment of minority students in medicine, at least on Stanford's campus. Well, I'm going to put that behind me. And the fact that we didn't have very many minority students, my class was only 86, and there was a handful of minority students. I needed to be creative. I had the very good fortune in my last 2 years of medical school to live in a Jewish house. And these are two of my Jewish roommates. They're two of my best friends from medical school. Daniel Kraft's in the middle. Lee Sanders is on the right. I'm the godfather of Daniel's son, and you can tell that they're Jewish and I'm not. And the reason I show this slide is actually for the trainees, because it's easy, if you're coachable, to find mentors who don't need necessarily look like you, act like you, or your same gender or sex, or come from where you're from. The fact is, if you can be coachable, you'll thrive. And I learned that trick very early on from wrestling and from my experiences in medical school. I was very fortunate to land at the Brigham and Women's Hospital in Boston. And I was very fortunate to have a wonderful program director, Marshall Wolf, who selected me to be one of 42 individuals in his residency program. And Marshall encouraged me to find a way to merge personal passion with academic rigor. Marshall Wolf had an open door policy, and he mentored me on a weekly basis. And he tried to figure out what drives me. And I told him, I was really interested in ethnic disparities, but I spent 3 years at Stanford in a genetics lab studying pulmonary disease, but I didn't know how to converge these two. I did not know how to make them overlap. They almost seemed antithetical. And Marshall was quick to point me in the direction of Jeff Drazen. Jeff Drazen at the time was the division chief of pulmonary and critical care at the Brigham and Women's Hospital in 1997. Jeff was studying asthma, genetics, and pharmacogenetics. He was a nice guy. He encouraged me to study asthma genetics, gave me a book, and I read the book, and I actually turned him down. And I said, no, I'm not interested. I really want to do health disparities. Jeff put his hand on me and said, son, I think you're making a mistake. Give it a shot. So I worked with Jeff for a year. He doesn't recall, but he sent me to the Harvard School of Public Health for training in epidemiology and biostatistics. And then I joined his lab later on. But because I already had genetics training at Stanford, I was able to hit the lab running. And we had a first author publication before I finished my residency at the Brigham. One of the great things that Jeff did for me, though, is he sent me to the American Thoracic Society, which is our professional society. I was one of the only medical residents there out of a population of about 20,000 individuals. But I challenged myself to look at all the abstracts. And one abstract that was published by the Center for Disease Control was this abstract. It showed Hispanic asthma mortality in the United States by geography. And what you can see on the left is asthma mortality and prevalence are highest in the Northeast, lowest in the Midwest, the South, and the West. And to me, this was key because I knew exactly what this was. This is my aha moment. But I want to tell you, this is where diversity comes in, because 1 million people looked at these data, but when I looked at these data, I knew what it was. And it takes a Mexican kid from California who studies genetics, who lived in Boston, to figure this out. I told Jeff that is not about geographic region. It's about Mexicans versus Puerto Ricans. And 2 years later, the CDC updated their paper and demonstrated that, yes, indeed, that it was Puerto Ricans versus Mexicans. But by 1998, before I finished my residency, we were off to the races. Jeff Drazen, Ed Silverman, and I came up with this idea to study the genetics of asthma in Latino Americans, or the GALA study. The plan was to recruit 700 Mexican and Puerto Rican family trios from New York City to San Francisco. Jeff was able to tap into the NHLBI funded asthma clinical research, and we were funded before the time I graduated from my residency. So to sum up the score at that time, I had, as a resident, I had a first author publication. We had funding to start de novo recruitment. But the way the match worked at that time, you matched 2 years in advance. And the wheels were in motion for me to come to UCSF, and in particularly to San Francisco General. And there I met some wonderful mentors. Dean Shepard took me in, even though he didn't study genetics of asthma, but he was a wonderful mentor and a wonderful coach, and has mentored me since 1998. Talmadge King was my department chair. He's a pulmonologist. He also took care of my mother, who was diagnosed with interstitial lung disease. And Talmadge helped me make connections all around the United States and Mexico and Puerto Rico. Neil Risch is probably the most world's famous population geneticist, took me under his wing in 2000. Kathy Giacomini became my department chair in 2004, and she studies pharmacogenetics. So we had a wonderful team of 4. Well, during my fellowship, it was time for me to implement the GALA study. And I have to say, it was very difficult. Number one, we were trying to get intact families. And that was difficult because the divorce rate is very high. So we couldn't recruit intact families from New York. And then number 2, Mexicans don't go to UCSF Parnassus Hospital. So I had to retool, and I spent every weekend of my clinical fellowship going to small clinics all around the Bay Area that served Latino patients and recruited Mexican-American families, mom, dad, and a child with asthma. Recruitment was going slow, so we expanded with the help of Talmadge King, who introduced me to Moises Selman in Mexico City, introduced me to Antonio Anzueto in Texas, who introduced me to William Rodriguez, and then subsequently Jose Rodriguez. And I'm very proud to say that by the time I finished my fellowship, my pulmonary critical care fellowship, we completed recruitment of 701 complete family units, Puerto Ricans and Mexicans. And with that huge data set, I joined the faculty in 2001. When I joined the faculty in 2001, I started what's called the UCSF Asthma Collaboratory. Collaboratory means that we only work with nice people. But the focus of my lab was that we focus on the majority minority, populations that have been understudied in clinical and biomedical research. We blew up the GALA-1 study. We've expanded all over the United States. We included sites in New York. We included sites in Chicago. We included sites in Houston. We included sites, continue with sites in Mexico City and Puerto Rico and the San Francisco Bay Area. And in 2013, I invited my first graduate student, Max Seibold, to join, even though he's not a clinical recruiter. Well, what we did, and I'm very proud of this, over the years, we recruited more than 13,000 minority participants. The majority of those individuals are children, probably about 10,000. All the people listed here are key principles that were involved. I mentioned Jose, Dr. Lenore, who was a president of the National Medical Association, helped me break into the African-American community. Neil Risch was brilliant statistical or population geneticists and epidemiologists. Ira Tager was my epidemiologic guru mentor. Louisa Borrell and Sean Exen helped me write the R01 that funded the recruitment of all these individuals. And all the individuals on the lower panel are the key principal people that helped me recruit. Combined, we created the largest and richest gene environment study of asthma in minority children in the United States. We collected very detailed information on genetics, clinical information, questionnaire-based information on perceived discrimination, socioeconomic status, and then geocoded measures of air pollution every single child. And one thing that we did right is we standardized spirometry at all the sites. We had every child, and this is little Juanito, breathing into a spirometer. And for those of you that don't know, a spirometer measures how tight your airways are. And we can measure drug response. So on the top, we have exhalation, and we use that as a quantitative trait to measure how severe someone's lung function is. Well, we were very fortunate because in 2016, the NHLBI provided us with funding to do whole genome sequencing on 16,000 participants, RNA-seq on 3,500 participants. Now we can integrate all this very rich genetic data with very rich clinical, social, and environmental data. And we have all that data in my lab at Rock Hall at Mission Bay. Well, it took a while to build this cohort. And so while we were building, we were doing other things. In 2000, the Human Genome Project, the initial sequencing was done. At that time, Bill Clinton, Tony Blair, and Francis Collins got up on stage at the White House, held hands and said, we're all 99% equal. Therefore, it should eliminate the idea of race and ethnicity. Many social epidemiologists and others jumped on this and said, this is a good opportunity to get rid of race in medicine for good. Well, I had worked with Jeff Drazen. I knew allele frequencies differed by populations, by race. So what I did is Elad Ziv, Neil Risch, and I got pretty upset, and we organized a coalition of scientists, not all of whom agreed with us, but we agreed to disagree and write about what we agreed on. And we made a strong, coherent argument, arguing for the inclusion of race, ethnicity in clinical and biomedical research. Well, I met a wonderful collaborator, one of my best in my entire career, Carlos Bustamante, about 12 years ago. And Carlos and I and Francisco de la Vega, we asked the question, who's benefiting from the Human Genome Project? Out of all the modern studies done in the world, who benefits? And what we found is that 96% of all modern genetic studies benefit European populations. Less than 4% of non-European populations benefit. Now, Europeans make up less than 16% of the world. So 84% of the world is missing out on modern advances in genetics. This is problematic, and it in fact pisses me off. Well, Alicia Martin, 8 years later, repeated our analysis and asked the question, who's benefiting from genome-wide association studies? Europeans are shown in red. You can see that there's been a persistent trend upward, that they're the biggest beneficiaries. And what she also pointed out on the bottom is that African Americans and Latinos make up less than 2% and 0.5% respectively of all modern genetic studies, even though we make up 38% of the US population. We are contributing a significant amount of tax dollars to the National Institutes of Health, and we are not getting our due diligence. All the money is focused on whites, and I think that's because the field largely is dominated by whites. Study sections are dominated by whites. White physicians, white scientists feel comfortable dealing with whites, and so it's easy for them to include whites. If we had more minorities, it would be easy for minorities to recruit minorities. Well, to that end, I want to add that the Human Genome Project systematically excluded Native American populations. So there are no biorepositories in the United States on Native Americans at all. And that's a travesty because that's a missed opportunity. I'm very grateful that a colleague of mine, Cheryl Winkler from the NCI, has assembled one of the largest biorepositories of Native Americans from Oaxaca, Mexico. She recently retired and asked me to be the steward of her samples. My lab accepted all the genome-wide or genetic data, all the plasma, but we didn't have the ability to accept the lymphoid blastoid cell lines, the CBCs, and the Precision Medicine Initiative program led by Keith Yamamoto accepted them. Now, I hope that Keith and his colleagues will raise funds from private foundations to do whole genome sequencing and RNA-seq so that we can freely share all these data with UCSF investigators. Now, finally, this year, 2020 has been rough. We saw with George Floyd, Breonna Taylor, and Ahmaud Arbery that we continue to have racism in America. In fact, racism is woven into our fabric. Jim Crow is not dead. He just went underground. We, as a society and as a world, had a racial awakening. Also, because of George Floyd, many people tried to use this opportunity to argue, let's get away from race and ethnicity, just like Josh Adler advocated. And what Louisa Burrell and Elad Ziv and I did is we assembled a team of scientists and physicians, minority and non-minority, leading minorities, deans of universities, CEOs of companies that are all minorities. And we put out a position statement arguing that the importance of race, ethnicity, and genetic ancestry still is important. But what's different about this article compared to our first New England Journal paper is that we used empiric data from the 13,000 individuals that I recruited to bolster our point of view. Now, enough with the background. I want to go back to this slide. We recruited, 13,000 individuals. My colleague and I, Elad Ziv, and I've worked with Elad for more than 21 years. We've always talked about using genetic ancestry and trying to leverage it to scientific advantage. Now, Jeff Drazen, my former mentor, always advised me, think about clinical relevance. Who cares if it's not clinically relevant? Well, I'm a pulmonary specialist and we do pulmonary tests. And I showed you this slide earlier, this is Juanito performing spirometry. Well, when an individual performs spirometry, we have to compare that individual to a population average. And in the United States, there are only 3 population averages. And that's demonstrated here. These are data by N. Haynes. On the left, we have male, on the right, we have females. On the y-axis, we have lung function. The higher you are, the better. On the x-axis, we have age. And you can see that when you compare African Americans to African Americans in green, they have lower lung function values than Mexicans, than Caucasians. And so any one of you who has lung disease, and if you come into a clinic, you're going to get assigned to one of these groups. Now, what happens if you're Chinese? What happens if you're South Asian? You are going to get assigned to the Caucasian group. Now, there's another equation called Global Lung Initiative, or GLI, that also has race-specific equations, but they have a pureed version where they took all the equations from the different races and collapsed them and averaged them. And so basically, we're getting a whitewashed version of lung function. Well, This got thrown in my face when I was a clinical fellow working in the pulmonary function lab. I had a firefighter that looked just like this guy. We know his history. He is Nigerian and he's white. The question is, what would you call him? Is he black or white? I know it sounds like a joke, but it's actually very important. The police would call him black, and he'd have a high likelihood of being killed or incarcerated. But as a physician and as a pulmonary specialist, I had to make the distinction. And I asked him, since I'm from the hood and I can code switch, I said, hey man, what are you? Are you a brother? Are you white? And he goes, I'm half. And so when the technician was doing this test, the technician was going to pull downs. You're either white, you're black, you're Mexican. But I knew from my own previous work search that African Americans are racially mixed. And this is a slide of about 300 African Americans. Each bar represents an individual who have genetic ancestry. Green, yellow, or orange is African, proportions of African ancestry. Green are proportions of European ancestry. And you can see it runs the gamut. That with going from left to right, you have decreasing amounts of African ancestry. So Obama might be the last to the right, he's half, but Denzel might be over there to the left. So with my colleagues, we asked the question, should we use self-identified race versus genetic ancestry? And here's what we did. We were able to access 7 independent cohorts of African Americans that are primarily healthy and had lung function. And we had 2 asthma cohorts that had asthma, lung function, and had genome-wide estimates of genetic ancestry. And what we found is that there is an interaction. The y-axis is lung function. The higher you are, the better. The x-axis is increased in amounts of African ancestry. And what you can see is it's an interaction. And when we wanted to compare an individual patient to a population average, there are only 2 in this case. There's a white reference equation, which would have been comparing your individual number to, say, 4.5 liters. Whereas if you're African American, we would compare your individual values, dependent upon your ancestry, to an African American standard at 3.9 liters. Now, mind you, that's a 1 liter difference. And on average, we have about 4 liters in our chest, and women might have about 3.5, 3 liters in their chest. But what we demonstrated is that dependent upon your genetic ancestry, if you're under the average African ancestry, which is 78%, we would underestimate your disease. If you are over, we would overestimate your disease. And what we did is we demonstrated that by including genetic ancestry into the equation, we can improve the accuracy of the diagnosis of lung disease by as much as 15%. That is huge and has tremendous clinical implications. Now, this is one of my favorite publications, my favorite collaborations. So I'm 55. I met Carlos Bustamante when I was 44. Carlos Bustamante is a professor at Stanford. He's now at Miami. But for the first time in my career, first time, I was 44, I met someone, a peer, who was interested in the same things that I was interested in, genetics, Latinos, bilingual. And what we did is we assembled an all-star team of Latino physicians and scientists. And I'm going to call Chris G. New, who is one of my PSBJ graduate students, an honorary Latino. But we had Andres Moreno Estrada, who is a Mexican physician, MD, PhD. Josh Gallanter, who is also Latino, who currently works at Genentech, was a postdoc in my lab, but he's from Venezuela, fully bicultural and bilingual. And what we were able to do is we were able to collaborate with the Genomics Institute in Mexico and anthropologists in Mexico and access indigenous populations Some people might call them tribes, but we just call them populations. And on the left, we did an ancestry-specific principal components analysis and demonstrated that populations, the indigenous populations, are just as genetically distinct from each other as Asians are from Europeans. And then we extrapolated that to contemporary mestizo populations in Mexico. And you can see that depending upon the ethnic subgroup on the left, the contemporary Latino population lined up to different geographic regions. And finally, what we demonstrated was, why is this important? So we had lung function on 2 Mexican cohorts of children. We had our Gala 1 study, we had the Mexico City asthma study provided by Stephanie London. And what you see in the bottom right corner is that the type of Native American ancestry varies, your lung function varies on the type of Native American ancestry based upon where you're from. So if you're from Sonora, you had high lung function. But if you're from the Yucatan, which is in the Southeast, which is where I got married, you had low lung function. And so instead of going from global ancestry like African, European, Native American, we went to subcontinental ancestry, showing types of Native American ancestry influence lung disease by as much as 10%. And what this means is that a decline of there's a northwest to southeast gradient. Depending upon the type of Native American ancestry you have, your lung function will drop by 10%. And that corresponds to a 10% decline in lung function, which corresponds to getting older by 10 years and having your lungs decline over 10 years. So what about social and environmental factors? We always get this question. I had a great postdoc in my lab, Maria Pino Llanes, who was a geneticist, now faculty in Spain. Nita Tucker was a fellow, who's a pulmonary physician, now faculty. And we had 5,500 Latino children. We asked the question, what's more important? Is it genetic ancestry? Is it environmental factors like secondhand pollution, air pollution, or socioeconomic factors, income, education, discrimination? And what they did is they demonstrated that ancestry was the biggest driver of lung function, at least in Latino children. And Maria went on to demonstrate that on the y-axis, we have lung function. The higher you are, the better. On the y-axis, the y-axis, lung function. On the x-axis, we have increasing amounts of African ancestry. And she replicated our New England Journal results. Now, these are all association studies, and I know that people here will want to see functional work. Well, we had the whole genome sequencing data, and Eunice Lee, who was a postdoc in my group, took this to the mat. She demonstrated that she could find that a chromosome 1q32, if you've inherited African ancestry, at that particular locus, you had a 0.2 liter decline in lung function. And she used this to identify novel genes, particularly TMM9 or TEM9. And she looked at rare variants. And what she found is that gene expression of TEM9 corresponded to gene expression of pro-inflammatory cytokines, IL-6 and IL-1 beta. Now, when you look at the alleles at that particular locus, if you inherited 2 African alleles, you had lower TEM9 gene expression than if you inherited 1 allele or 2 alleles. The same is true for the other variant that's in LD with them. Eunice went on to demonstrate that amongst Native Americans, if you inherited a Native American segment of chromosome 5q35, you had increased lung function by 0.15 liters. And these are clinically significant values. So what does this mean? We're talking about disease misclassification. If we throw out race and ethnicity, it's like throwing out the baby with the bathwater. We get disease misclassification that has clinical implications, disability ratings, lung transplant referrals, preoperative risk, workers' comp, impairment. Now, I want to remind my pulmonary colleagues who have been reluctant to integrate modern technology into pulmonary medicine. I'll remind you that penicillin was created in 1928. The Human Genome Project was completed 21 years ago. Don't you think it's time to improve and integrate modern advances in science and medicine into pulmonary function testing? I think it's time. I think it's bad if we go back to 2000 and say, let's whitewash everybody and use one size fits all. I think that's equally discriminatory. Now, there have been a lot of discussions on this topic. There have been editorials written. The ATS has had special sessions on this. And I could tell you, having been a participant in those committees, 90% is driven by commentary or viewpoints or opinions. Less than 10% are empirical evidence-based studies. And I'm very proud to be able to say, to the best of my knowledge, we were the first in the world to correlate genetic ancestry with clinical measures like lung function. And of that 10%, my lab and my team has contributed the most. We have several copycats who have also supported our work, but the fact remains that we are making big decisions, just like Josh Adler did, on very little empiric evidence-based data. So the takeaway that I want you to take from this point of the lecture is that race, genetic ancestry, and medicine are intertwined. The epidemiologic importance of race and ethnicity will never go away. Throwing out race and ethnicity without careful clinical consideration is not the answer and may lead to more inequalities and increased health disparities. What we need is empiric evidence and not opinions. So I want to switch topics and I want to talk about some fun stuff that we're doing. This is brand new, hot off the press. We know that 80% of disease-associated variants lie in non-coding regions. Now, we have some of the world's largest data, whole genome sequencing data, RNA-seq data. And this is a project that was led by Angel Mock, Linda Kachuri, supervised by Elad Ziv, and also helped by Donglei Hu. What Angel, Linda, and the team did is they took our whole genome sequencing data from African Americans, Puerto Ricans, Mexicans, other Latinos, combined it with RNA-seq data from the same populations. And what they demonstrated was that gene expression patterns differ by race and ethnicity. This paper is under review. It's favorably reviewed at a high-profile journal, and you'll have more to come. But they demonstrated that the gene expression heritability tracks with genetic ancestry. This slide is a little bit complex, but I tried to make it as simple as possible. On the left, I have a dimmer switch, African ancestry. On the right, I have the proportion of phenotypic variation from the heritability of gene expression, and whether or not it's due to genetics or environment. And what Angel and her team demonstrated that with increasing amounts of African ancestry, the proportion of phenotypic variation in the population due to genetics increase. And that's likely because Africans have more genetic variation than any other population in the world. What she also demonstrated was that 30% of the genes, this is the whole genome now, have at least one ancestry-specific EQTL. Now, a new analysis that Angel and Elad did is they try to take a crack at the heritability of asthma. We know that the heritability of asthma from twin studies ranges from 35 to 70%. But those twin studies are done in whites. And so it's practically irrelevant to a cosmopolitan population like the United States or the other 84% of the world. So the way for us we as a scientific community to get at the heritability is to do genome-wide association studies, which look at common variants. Common variants have only been able to explain 13% of the heritability of asthma, but using whole genome sequencing data, Elad and Angel were able to increase that to 53% by including rare variants. Rare variants make up about 70% of the heritability of asthma. And this is in minority populations. This is a first, this will be a groundbreaker study. Now, I want to return to my roots. I didn't crack this nut. I didn't explain why Puerto Ricans have higher prevalence than Mexicans. I will tell you an aside, since I have a minute, that Sam O published a paper demonstrating that 95% over the last 20 years, 95% of the NIH research focused on lung disease has focused in on whites. Even though minorities represent 38% of the population, minority children today, born today, more than 50% are non-European. So I wanted to start a longitudinal study. And I asked my peers to help me. One of the ways we did it is we looked at the early origins of asthma. Sam O published a beautiful paper demonstrating that viral infections were the strongest risk factor for the development of asthma, particularly RSV and human rhinovirus. Now, I got a grant to do more recruitment in Puerto Rico in 2013. I invited Max Seibel. Max was my first graduate student. He was a faculty at National Jewish. He was interested in nasal epithelial cells. So I allowed him for the first time to come to Puerto Rico. And he brought a nurse that showed us how to do the nasal epithelial cells. And the clinical team in Puerto Rico was able to do that perfectly. And I'm showing this to show you how it looks uncomfortable, but it's not that bad. In fact, on the lower right corner is my colleague Noah Zeitlin, who got a nasal brushing. So I'm going to put this in context. The only way pulmonary physicians get lung tissue is to do an invasive bronchoscopy. Now that's pretty invasive. You cannot do it in children because it's unethical. We had a whole system on gathering nasal brushings. And in fact, I paid for the collection of 1,000 nasal brushings for Max to jumpstart his lab. And what Max did is he demonstrated that the gene expression patterns in the nasal genes overlapped 90% with bronchial gene expression patterns. So it obviated the need for us to do bronchoscopies or the tremendously invasive bronchoscopies. And now, because the nasal brushings are so easy, we can get thousands and thousands of brushings in a couple of months. And what Max was able to demonstrate is that there are different flavors of asthma based on gene expression patterns of the nasal epithelial cells. He demonstrated that there's Th2 high, Th2 low. And that was critical for going forward. Now, this is Jose Rodriguez Santana. We have been working together since 1998. He's been a father figure, a mentor to me, a colleague. We worked together for many, many years in many different settings. Jose happens to like fishing. I like fishing. We brought in our closest collaborators, Maria Pino Llanes, who is one of my postdocs, who's now a faculty in Spain. She's a geneticist. We brought in Luisa Borrell in 2004. She's a social epidemiologist at the City University of New York. And because we study minority populations, we needed to have social epidemiologists. We needed to have geneticists that were proficient in understanding racially mixed populations. Then I re-invited Max back into the team. Now he's a professor at National Jewish. And here's a scouting trip that we did. Max is on the left in the red t-shirt. Sam is our lead epidemiologist, who is the designer of our longitudinal study. And we took time out and went fishing, and that's my big fish on my 50th birthday. But on a serious note, we had a planning meeting in Puerto Rico. This is the back of the envelope meeting, and we are planning a longitudinal study, which ended up being a birth cohort study. And we came up with this, Primero research. Primero is Puerto Rico's first birth cohort study. It means Puerto Rican infant metagenomic epidemiologic study of respiratory outcomes. I paraphrased a line from Abraham Lincoln's Gettysburg Address: science of the people, by the people, for the people, shall not perish from this earth. We are fortunate that the NHLBI funded us. Our goals were to recruit 3,000 mother-infant pairs, follow them for the first 5 years, identify the early origins of asthma, and study the life course of health and disease. Now, anyone who's in science and medicine knows the Framingham birth cohort, or Framingham cohort. Primero is a birth cohort. We could do so much with it. And I hope that this cohort outlives me. We are collecting data on mothers. We can look at the epigenetics of maternal stress. We are collecting data on infants, cord blood, nasal brushings. We can look at genetics, gene expression of the host, the viruses, longitudinal epigenetics, disease biomarkers. We are collecting longitudinal data on the children, looking at the long-term impacts of genetic and environmental risk factors on health, gene expression, and epigenetics, not just for asthma, but for obesity and a variety of other diseases that come over the life course. Now, this is an example of a postpartum mom, 2 days postpartum, bringing her baby to the clinic. This is Dr. Rodriguez Santana doing a nasal brushing. And we started recruitment in March of 2020 during the COVID shutdown. And I'm very proud to say that during that time, we've recruited over 1,000 mother-infant pair dyads despite the shelter in place. This is a testament to Jose and his team. So I'm going to end the science there. I covered a lot of ground. I'm a complex person. I think my worst experiences were growing up as a single parent to a school teacher. All the things that people talk about, adverse childhood experiences, I experienced. We had food insecurity. We were poor. I missed Christmas. One day, my mother gave me an IOU and started crying. She told me, I'd rather see you dead than on welfare. I had the disadvantage of being a Latino kid from the hood. I had grit because I was born poor. I learned how to fight. I learned how to wrestle because I needed to in a gang-infested barrio. I was very fortunate to get into Stanford. And not only Stanford, I was very fortunate to get into the medical scholars program where I did 3 years of basic research on lung disease, genetic research on lung disease. It was on transplants. And transplants are for wealthy people, primarily white people. And they didn't address my interest in health disparities. So I dropped it quickly when I got to Harvard. Harvard was great. Harvard, Marshall Wolf bred social activism. People like Paul Farmer, who started Partners in Health, Jim Kim, who led the World Bank, the current Surgeon General, Vivek Murthy. I was very fortunate to get into UCSF, where there's a strong role of activism. And as a pulmonary physician working at San Francisco General, I was there during the AIDS epidemic, and I saw the activism, not only on the physicians, from the scientists and the community. I was very fortunate to get a scholarship from the Robert Wood Johnson Foundation to go back to UC Berkeley and improve my training in epidemiology and biostatistics and create the idea for the GALA-2 study. Now, it's time for me to take a few minutes just to thank the people that supported me. And one of my biggest supporters has been my wife. We met in college when I was a sophomore. She started dating me when my stock was low. I couldn't even give away my stock. And here's a picture of us. We got married on Chichen Itza, which is the Mayan Empire. The Mayan civilization were some of the greatest mathematicians in the world. They created the zero. We produced two beautiful daughters, and I wanted them to be smart, independent, self-confident. My daughter Milena on the left is a project manager at Microsoft. She's a computer scientist. My daughter Maya on the right is a computer scientist still in college. She is named after the Mayan Empire. And I am proud to be able to say that I delivered them both. I delivered Milena at the Brigham and Women's Hospital. I delivered Maya at UCSF. Now, going back to my family life at the lab, even though I'm getting the opportunity to present today, I have hidden pillars of stability. And as a fatherless child, always wondering where I was going to get food from, not having Christmas gifts, I like stability. Celeste Ng, who's there on the left, has been the steel girder of our lab, the backbone of our lab. She's going on 17 years in my lab. Sandra's going on 16 years. She's our clinical recruiter. Dong Lei and Scott are going on 14 years in my lab. Jennifer Laraway is one of my right-hand persons in helping me write all my grants and papers. I want to thank my team. I especially want to thank Neil Risch for taking me under his wing. Elad Ziv for being my partner for the last 21 years. Louisa Borrell being my partner on social epidemiology and everything since 2004. Noah Zeitlin, who is one of my first hires. I want to thank the Sandler Foundation for providing the fuel that helped me go forward, and my very diverse team in my lab. I want to thank my funders, and I want to say something last. We all start off as students, and then we become trainees and postdocs, and then we become assistant professors. And it's like wrestling. One day you're in the mat, one day you're on the side of the mat and you're coaching. And I'm very proud that I've left a strong and proud legacy of trainees, most of whom have gone into academics, academic medicine, or have scientific careers. Some of them will be world leaders. I think Christian Yu will be the next world leader in population genetics. Same with Noah Zeitlin. And many others will have a significant impact. Josh Gallanter has a tremendous job at Genentech, a leadership role at Genentech. Leslie Samity just went on to the NIH, and she's now my program officer. Angel Mock is going off to do great things. Ryan Hernandez was my first hire at UCSF, and Max Seibold is doing very, very well. So I'm going to end there, and I just want to say thank you for the opportunity, and I'd be happy to take any questions.
Wow, Dr. Burchard, thank you so much. I so wish that you could hear the very loud applause from the almost 200 people on this call, which if we were in person, I think at this point would probably be a standing ovation. You're experiencing the deafening silence of Zoom right now. And I just hope to convey our thanks for your extremely inspirational talk, hearing about your life course, your scientific insights and advancements has had a huge impact on the audience, I know. We have a couple of questions in the Q&A, but first I want to just introduce Elad Ziv, who you mentioned many times, and I think he's going to kick off the Q&A. Dr. Ziv is a professor in the Department of Medicine, and I think I maybe saw him on one of the fishing boats, but I'm not positive. So
no, I should introduce him.
Okay, please do.
I'm sorry, that was my mistake. Elad and I started off together in, I think 2000, maybe 2001. Elad's a general internist. He's trained in statistical genetics, and we made a pact. We knew that the likelihood of academic success was very low. And I told Elad, we could swim alone and have a high risk of failure, or we could swim together and have a high risk of success. And that was in 2000. And we worked together closely from 2000 to now. We drove twice a week to Stanford to take a course with Neil Risch when he was a faculty there on population and statistical genetics. We share a lab. We recruited together. We've gone to Mexico and taught classes. I helped connect him to collaborators in Mexico to jumpstart his breast cancer and Latinas cohort. And it's been a wonderful collaboration, and it's very synergistic. And I'm very proud to know him and to be his friend and call him my colleague.
Thank you. I just want to say also, I want to thank you for being my teacher for these 2 decades and going strong, and also highlight the way I think that you have done your science, both with the combination of determination and passion. I think that's really been the things I've learned the most from you. I have the prerogative of the first question. So I'm going to ask you kind of a broad question, which is genetic ancestry in medicine. Where do you think it's going? Do you think we need to be measuring genetic ancestry? You think we should be measuring? How do we incorporate it? It's become something that many people now know about just from their kind of recreational genetics. But is this something that you think we need to kind of put into medicine? How?
Well, I actually think that I'm only going to talk about lung disease because I'm a lung specialist. For the majority of patients, it doesn't really matter, probably 70% of patients. But in those few patients that go, say, to Parnassus, where we need a specialty diagnosis, and what we demonstrated in the New England Journal paper, that there's a 15% error rate. And that 15% error rate could put you on one side of the cutoff for whether you qualify for transplant or not, qualify for disability or not. And so in those special cases, I would use genetic ancestry we talk about precision medicine, but the way my field, pulmonary field, is going is that we're going back 30 years to a white-only reference equation. And like I said, penicillin was developed in 1928. It works. It's time to get with the speed. The Human Genome Project was completed 21 years ago. It's time for us to get up to date and start incorporating modern advances in medicine. You know that the cancer field has been revolutionized by genetics. In fact, God forbid that any one of us gets cancer, but if we do, if you're at UCSF, your tumor is going to be biopsied and sequenced, and we'll be able to determine what drugs work for you and what drugs won't, and we'll be able to subphenotype your cancer into various types that will respond to treatments.
Elad, should we go to some of the questions now? We've got.
Sure. Go ahead, and I've got more questions, but let's kind of let you moderate.
Great. I'm going to ask you some of the questions that are coming in on the Q&A, and I see some celebrity sightings in the Q&A too. So actually, maybe I'll start there because I see Jeff Drazen has put a question in. I don't know if you knew that he was listening. So his question is, how reliable is assessment of genetic ancestry, which he puts in quotes? Is the technology used to make an assignment now stable, or can a person have their genetic ancestry evaluated and placed in their medical record for use as an immutable trait?
Well, thank you, Jeff, for showing up. You said it was you weren't going to show up because it was too long. So I'm very pleased that you're here. It is an immutable trait. We don't have complete reference equations for the entire world. And so that's a shortcoming, but we do have very good reference equations for the majority populations in the world. And that's why I'm glad that UCSF accepted the Oaxacan samples. There are no biorepositories of Native American samples except for what exists in Mexico, and now UCSF has one. The genetics field is, and National Geographic are continuing to collect samples all around the world. And so the genetic estimates will get further and further improved. And if you've signed up for 23andMe, every now and then you get an update where your estimates get a little tighter. And that's because they're adding more reference equations to their data set. But yes, it's an immutable trait. Just measure it once and you're done.
Great. We have a lot of questions coming in, and please keep sending them. I also was just told, I want to say that this is the faculty research lecture series apparently with the highest recorded attendance in a long time. So again, I think it just speaks to the importance of your work and the engagement that the UCSF community has with it. We have a question from Neil Poe saying, does the correlation of ancestry with lung function parameters represent biology or environmental, like social and behavioral causal influences, since self-identified race is associated with ancestry classification? Of course, social effects can have effects on biology, e.g. epigenetics.
Sure. So we did publish that very nice paper looking at the social and environmental factors that we collected. Now, being an epidemiologist, I know that we cannot collect everything. So we demonstrated, even after adjusting for social and environmental factors, genetic ancestry was the strongest predictor. One of the projects that I didn't have time to talk about was a very cool paper published by Josh Gallanter and Noah Zeitlin. Everybody knows that epigenetics is influenced by social, environmental, as well as genetic determinants. We had data on hundreds of Latino children, different types of Latino children. We had genetic ancestry, we had methylation, we had social and environmental determinants. And what Josh and Noah demonstrated is that 75% of the methylation patterns are driven by genetics, whereas 25% are driven by social and environmental factors that co-vary with race and ethnicity, which was one of our best papers because it tells us that we have a long way to go to really disentangle what's more important, genetics or the social determinants of health. Now, the social determinants of health, I can ask you this question. What's the likelihood of a kid dying in Ferguson, Missouri? Okay. Your first principal component would be male or female. Okay. And then age. And then race. That has nothing to do with biology. That is purely a social determinant of health. And we are seeing that over and over and over again. We have a lot of work to do. But unfortunately, the study sections are primarily 85% white male, and they don't appreciate this. And we get ridiculous reviews when we study minority onlys, and we get reviews, where's the white reference group? How is this generalizable to whites? And that is discriminatory because when we have white studies, and when I was on study section for 6 years, when a white study came in, we never said, where's the black reference equation? Again, as I said, racism is baked into our system. Jim Crow never went away. It just went underground. And we saw it rear its ugly head in 2020.
That's a perfect segue to a question from Bob Wachter that's asking you to take on some political environment issues. So his question is, I'd be interested in Esteban's take on how the political environment in which all matters of race become potential hot buttons influences his work, particularly his argument that we don't necessarily throw out race as a meaningful variable on certain measures?
It's a delicate topic. But like I said, I've grown up with race all my life. My mom was dark skinned, spoke Spanish, went to a segregated school all the way up to high school. My whole family is dark. Spanish is their first language. I see it every day. And it's difficult, but I'm very comfortable talking about it.
Well, I want to give you a recognition. Then there's some things coming in on the chat, and I just want to read one from Raul Rodriguez Jr. It says, "Thank you, hermano, for allowing me to be part of this today. You've made all of the kids from the mission overly proud. I say this with tears of joy. I love you, brother." I just wanted to read that to you because I don't know if you'll see it after. And then I wanted to put this question, which comes from someone from the University of Iowa, who actually wrote "Go Hawks" because wrestling season is starting soon. But this is some mentorship issues. So it says you've done a wonderful job training the next generation of physician scientists, but can you share some of your thoughts about how we can get underrepresented or historically excluded faculty in the room for funding and editorial decisions?
So we have no problem recruiting for the NFL and the Major League Baseballs. So there are new ways to recruit. And I think that we have to have a holistic view. And I think that UCSF had the largest number of minority students, underrepresented minority students in the medical school in this entire history this year. So there's a way to do it. And we just have to think out of the box. Diversity adds scientific value. It adds different ways of thought. My experience at Harvard, looking at those data, and I told you it takes a Mexican kid from the Mission District trained in genetics who moved to Boston to know that that wasn't geography, it was Puerto Ricans versus Mexicans. People talk about the pipeline, but I don't agree. And people tell me to be patient, and Dr. King talks about being patient, but 43 years is a lot to be patient for. I'll be dead in 43 years probably. So I think we have to rush on this.
I think we have time for maybe one more question. This is from, I believe, your colleague, Carlos Bustamante.
Mm-hmm.
And so, but this is about the future. So said, thank you for your great talk. The question is, as you think about the next decade, what role could a Latin American biobank like the UK Biobank play? And how could pharma help move this field forward? How can we advocate this for non-NIH funding since they've continued to miss the boat on this issue? And he says, to me, NHLBI has been uniquely inspired institute, but the rest is, quote, too little, too late. So thinking about the next decade.
Latin America represents 18% of the world population. It is incredibly diverse. And they have been tremendously untapped. Even in the United States, we have the Study of Latinos, SOUL. They recruited 16,000 Latinos. I was the one that helped design it with Francis Collins, but it's locked up. Very few people can get access to it, so it's almost useless. The Million Veterans Program, same thing. Tremendous resource, but it's locked up, almost useless. Kaiser, beautiful data set, locked up. We need to have increased diversity. Latin America is a tremendous opportunity, untapped. It's in our backyard. We have plenty of scientists that are bilingual, bicultural. I've had Josh in my lab who can do that, go to Latin America. Maria Pino Llanes, Puerto Rico is another example. It's tremendously diverse. And the admixture adds benefit, because within one population, you get three. And you can look at European, African, and Native American ancestry.
Great. I think we're going to have to end, unfortunately, but I did want to highlight that Daniel Kraft is putting in a comment, and he did bring up what looks like a nickname of Stubby. Am I saying that right?
So in the Jewish house, they would call him Stubby, yes.
And so he is saying hello and asking about including your work into medical education and how it can impact clinical care and medical education. So maybe we'll have that as the last question.
Well, you know, the Bridges curriculum is trying to standardize what we teach. And I don't believe in censorship. I have a pretty good career on showing that there is biologic benefit to looking at diverse populations. And I'd hate to be censored. So I think that we can have a variety of point of views in the medical education, not only in the School of Medicine, but I taught pharmacogenetics for about 14 years in the School of Pharmacy, and we talked about this all the time. In fact, we tested the children, not children, the students. We performed pharmacogenetic testing in all 99% of the kids. Sam O and Alan Wu and I did. And with that, we could show that there's racial differences in gene frequency for pharmacogenetic variants. And that plays a role in who responds to Plavix, who's going to respond to tamoxifen, who's going to respond to XYZ drug. And that all corresponds to race.
Great. Well, I'm sad that we have to end. I just, on behalf of the Academic Senate Committee on Research, want to thank you so much for incredible talk, scientifically inspirational, but also just inspirational to so many about your life journey and successes. And a reminder to everyone on the call that this talk will be available on the Academic Senate website. So please watch it again and have your colleagues and trainees watch it as well. And thank you so much, Dr. Burchard, and congratulations.
Thank you very much.